In the first two lectures, we reframed what a cell is and why boundaries and compartments are necessary. But boundaries alone are not enough. Once a cell is compartmentalized, a new problem appears. How does the cell coordinate what happens across those compartments without losing control? That problem is solved by signaling, but not in the way it's usually taught. Signaling is often introduced as information transfer. Signals are described as messages that tell the cell what to do. That framing sounds intuitive, and it hides the real danger. Cells are not static systems. Nutrient levels change. Damage accumulates. Compartments experience different conditions at the same time. If each part of the cell acted independently, the system would quickly tear itself apart. Coordination is not optional. It is required for survival. Cells are not trying to understand their environment. They are not interpreting messages. Signals are better understood as control inputs. They bias the system towards certain actions and away from others. The goal is not accurate representation. The goal is coordinated behavior under uncertainty. Once signaling exists, a new risk appears. Signaling systems don't just coordinate action, they amplify it. Pathways amplify small inputs into large responses. That's necessary. Otherwise, signals would be lost in noise. But amplification is dangerous. A false signal can trigger inappropriate growth, unnecessary arrest, destructive elimination. Once these processes begin, they are difficult or impossible to reverse. Because signaling is dangerous, restraint is essential. Cells tolerate uncertainty. They delay commitment. They require confirmation before acting. This is why signaling systems look slow and redundant. They are not inefficient. They are risk averse. Cells manage signaling risk using three strategies: location, feedback, and integration. Where a signal occurs matters as much as what the signal is. The same signal can have very different effects depending on which compartment it reaches. Location determines which targets are accessible, how long the signal persists, which safeguards apply. Spatial control is not a detail. It is a primary regulatory strategy. Signaling without feedback would be catastrophic. Once activated, pathways would continue unchecked. Feedback mechanisms shape signal strength, duration, reversibility. Many forms of cellular dysfunction reflect failed feedback, not missing signals. No single signal is trusted on its own. Cells integrate multiple inputs before committing to dangerous actions. This slows response, but it dramatically reduces catastrophic error. Safety matters more than speed. Even with all this control, signaling is never precise, and that's not a design flaw. At the molecular scale, randomness is unavoidable. Cells don't eliminate noise. They build systems that function despite noise. Robustness, not precision, is the design goal. Once we strip away the metaphors, signaling looks very different. Signaling exists to coordinate actions that could destroy the cell if misapplied. It limits escalation. It delays commitment. It manages risk across space and time. This is why signaling failures often look dramatic. They involve systems designed to operate near the edge. This is the key idea. Cells don't signal to learn, they signal to decide. Signaling is about timing, restraint, and coordination, not information transfer. Signaling is in place. The next question becomes unavoidable. How does a cell decide when to grow, divide, or stop entirely? That's where we go next.